Thank you very much. So we have a second talk now for Fred Adler talking about a whole variety of fascinating topics. So I don't want to take his time.
Okay, well, I'll try to follow Alan's lead in keeping this short and touch on a few things. So I was first going to say a few words about some rhinovirus work we did. So rhinovirus is the real cause of the common cold, with coronavirus as being a distant second in my view. Then talk about this class I'm teaching very briefly, just in the spirit of multi-scale modeling, and then finally try to present my overview of how I'm thinking about this whole field, which should, I hope, elicit some discussion. So rhinovirus is a great model system. It's the most common cause of the common cold. It has a lot of really interesting elements I'll only touch on a few of, but in a way, it's a nice example of a well-adapted virus. And we wanted to use it to look at this whole realm of interactions between phenomena. So the first one, and this is something that Alan touched on, is obviously a great interest in transmission, is thinking about the links between transmission and symptoms. And so we had a data set on family transmission here in Utah, and you get this interesting result that if you count up how many people are infected within a family and ask how likely are other members to get infected, you see a strong relationship with symptomatic people and no relationship with asymptomatic, which really violated my initial thought. I thought asymptomatic transmission would be really important in rhinovirus. Apparently it is not. If there's no asymptomatic transmission, that means it's also an evolutionary dead end. The second piece to look at, which was fortuitously involved coronavirus, was looking at the transmission in relation to other viruses. The upper respiratory tract is a very busy place. That's why I started studying it, because I'm an ecologist at heart and I wanted to look at a diverse ecosystem. So among the many, many types of viruses there are both rhinoviruses and the 4 seasonal coronaviruses. And we found this interesting relationship. Maybe I'll focus on this one, is that individuals who had no rhinovirus recovered from coronavirus, that is, lost a concurrent infection much more quickly than individuals who were infected with it. And so currently one of my students is modeling the potential implications of this sort of viral interaction for the long-term epidemic. So if being co-infected means that you remain infected longer, you may also remain infectious longer. It's a fairly small data set, so we didn't have the power to necessarily detect all these interactions, but I think there probably are interactions going in both directions between these two viruses. The last piece from this that I'll talk about a little more is the interaction between transmission, immunity, and evolution itself. This tree, which obviously you won't be able to read any of, shows the rhinovirus serotypes, of which there are a ridiculous number. These are all start with the letter H, meaning these are human specialist viruses. Among these are two types, the major group, of which there are 90, and the minor group, of which there are 12, though there are many more. People just got bored counting when they got to 100. And so major groups enter cells in a different way from minor group. And in particular, the major group rhinoviruses use a particular receptor called ICAM1. And this has very interesting indirect effects that we did attempt to model, which basically reduce the adaptive immune response and extend the duration of infection. Minor group viruses use a very different receptor to get into cells and do not have this effect. From this, we can make a prediction, just from this sort of the immunology of this interaction, that minor group viruses, because they don't suppress the immune response, should elicit more immunity. And if they elicit more immunity, they should evolve more quickly. And amazingly enough, there were data, these are old data from, I think these were from the 70s even, I can't remember now, looking at antibodies, I think a year after an infection. And we in fact see this effect. The major group bars are taller because there's more of them, but the majority of major group rhinovirus infections in fact had no antibodies, and a majority of minor group rhinovirus infections have antibodies. Obviously, this is highly relevant to the SARS-CoV-2 pandemic, where we are quite concerned about the duration of antibody response. I should say that for both rhinovirus and the seasonal coronaviruses, we know that immunity is temporary. People get the same ones multiple times, but there is a duration of an immune response that differs between them. On the evolutionary side, we could then ask, do we see differences in the evolution rate of these sites? I won't go into details, but a colleague of ours developed a model to say which sites on the virus are exposed to the immune response, as opposed to what sort of a previous, more crude way of characterizing them as antigenic or non-antigenic. Using this nifty method of exposed sites, we could see that these bars, the blue ones for the minor group virus, are taller, meaning they mutate faster. And so, in fact, it looks as though, and this is a ratio of exposed to non-exposed sites, right? So, in fact, we do see the minor group viruses eliciting an immune response and running away from it, sort of like what we expect. All right, so here we have a set of relationships between transmission, immunity, and evolution that we hope sort of shed some light on this virus. So from this, we built a model of a level of complexity similar to what Alan described of the interaction between rhinovirus and the immune system. And somewhat appealingly, we could plot the results in a summary of sort of the amount of memory predicted on the horizontal axis here and the damage, the fraction of surviving cells on the vertical axis. Rhinovirus, typically, unlike influenza, it really leaves a lot of cells behind. This is why you do not get a horrible sore throat from it. So the major group of rhinoviruses we think look like this, quite successful, we all get them every year, produce very little memory and very little damage. The minor group viruses, though the evidence for this is weak, we predict are somewhat more virulent, but will induce memory because they do not slow down the dendritic cells, which are the cells that present the virus to the rest of the immune system. The model also predicts a possible type, which is really nasty, which would clobber you, make you feel as sick perhaps as you do with the flu, and shut down the memory. We don't know of such a thing. There was a recently discovered group of rhinoviruses, the HRVC group, which I was concerned fit here, but it seems like they don't. They seem to act more like this. But this shows that viruses have a huge realm, at least within the context of this model, of possible dynamics, and the ones we see make some sense. These black ones, as far as we know, are just intermediate cases. So I want to say one thing about the sort of broader context of rhinovirus within its genus, which are the enteroviruses. And so we've seen here a switch in receptor, which is between getting into the cells through one thing and through the other. So as we know, SARS-CoV-2 uses the ACE2 receptor, which is also used by SARS itself. So here's ICAM1 and the low density lipoprotein receptor hanging around here with the different rhinovirus species. But interestingly, rhinoviruses live within the enteroviruses, and luminaries among the enteroviruses include things like polio. So they're really extremely closely related. And we see a lot of this receptor shifting going on back and forth between things like ICAM1, other receptors, things unpronounceable receptors. So there's a lot of evolution happening at this level. Why this is important, I really don't know. It may have something to do with immune escape. It may also have something to do with shifting host cell tropism to be able to coexist with other viruses. Okay, so I put these on fast speed. So that's it for rhinoviruses. I just wanted to say a brief word about this class I'm, to quote, teaching at the moment. So I was signed up to teach the math modeling class, and then this pandemic hit, and so I thought, what the heck? I'll use this as an opportunity to educate the students about a bunch of different flavors of modeling, particularly of pandemics. So, so we started off with epidemiology, and then sort of extended this to structured population models, looked at dynamics of vaccines, some evolution models that are similar, though obviously simpler than the ones I mentioned, and then looked at some of the issues that Alan mentioned, dose response curves, which I think are extraordinarily interesting and important. Some very, very simple immune models, which actually had some of the character, again, of what Alan talked about. And then sort of moved out of my comfort zone of things that look like biology into the physics of droplets and aerosols, thinking about things that are much more economic, like the queuing theory that underlies the overwhelming of our hospital system that I could look out my window and see right now. And most interestingly, in a way for me, is thinking about the economic impacts. So this is a smallish course run partly in person. And a central part of this is the project. I thought it would just be fun to put up for a second the project ideas that the students came up with for this. And it really is a remarkably diverse set of topics, which I think really exemplify multi-scale modeling, right? So we have things here at the top that really look more like the human sciences, economics of the healthcare system and of toilet paper in this case, how attitudes move around, things that look much more like epidemiology, super spreading and so forth. And then some really interesting ideas students came up with. One is like trying to understand the devastating physiology of blood clots underlying this disease, the effects of, this is ski country, the effects of COVID-19 on people not being able to go to ski resorts and instead going to more dangerous places and injuring themselves because they're not equipped for backcountry skiing. And one student is looking at what the effects of this are going to be on the foster care system, or in a similar one in a way is how will natural disasters affect the spread. So to me, the students have done a great job, not just in challenging me, but in challenging themselves to think of this, of the power of mathematical modeling to unite across many different levels of organization, even outside that of biology. So the last thing I wanted to say, also really briefly, is attempt to put together what I'm thinking of is the COVID landscape, but you could really see this as a pandemic landscape. So I'm starting here with the arch of what I think of as a biology department's typical purview in basic biology. And this is what my department runs from biochemistry to evolution. And it tends to be ordered in this way, as if these things are getting bigger and bigger. But one of the things that really inspired me to work on rhinoviruses is that evolution is biochemistry. We just saw it, right? If evolution is changing receptors, changing receptors means changing maybe even a single amino acid. And this is known for the major and minor group rhinovirus. And so evolution wraps right around, and of course affects everything here. And for viruses, these supposedly different spatial scales are all happening exactly at the same time. And I'm hoping that becomes slightly more clear with the rest of my diagram. You can think of spin-offs, related fields to this sort of basic biology landscape. There's the human sciences that I mentioned. Human behavior, obviously crucial for viral spread. Economics, crucial for thinking about the consequences of different control measures. The sort of more related disciplines of the physical sciences, biophysics, which is quite close to biochemistry in many ways, physics itself, like that of aerosols and droplets, and then of course, the one ring to unlock them all, the mathematics and data science that unify these things. Then we're fortunate here in Utah to have a medical school that's on campus and is actually friendly to us. And so sort of this is a crude version of some basic biomedical fields, which include looking inside the body, and then these are things look outside the body, and I threw dose response in there because I needed a place for it. And then there's much more applied things, designing the drugs themselves, delivering the drugs, using the drugs. Okay, so the last thing I wanted to attempt here is, drawing arrows, which is one of my specialties, is to think of all the links among these. What I think many of us are trying to do is link the basic sciences up here at the top, which is where I live, with where many of our collaborators live in the medical sciences, though of course in this group we've got people, I think, who represent all of these areas. This list over here is the sort of basic science components that I extracted from our list of subgroups. And I'm not going to belabor this much, but I had fun trying to put numbers on these different arrows connecting these things. And this is how I see this landscape. I've worked in many of these areas myself. I think mathematical models are the exact way that we can unify them. And I think that this group is in a unique position to bring all of these forms of thinking together. We discussed briefly the other week, epidemiology in a way is special, partly because it has its own group and it's a very well-established area. But if you look at the connections that are made between all these areas, and I ran out of space to draw arrows, all of these more mechanistic pieces link back to what we, at least in the control of pandemics, care about, which is stopping pandemics with all the tools we have available, whether they're these biomedical tools or whether they're human tools, and understanding that through mathematical and biological tools. Okay, with that, I'll just thank the rhinovirus people, and then this is our CO4 group. And I'll go back to my fabulous diagram, and oops, I'm sorry to go through my arrows, and thank everybody for their attention.
Thank you very much. It's a great overview. A nice coda to Alan's talk. Time for a couple of questions.
I have a few, Fred.
Thanks.
What's the molecular basis for these rhinoviruses affecting dendritic cell behavior?
I don't remember the details. It's really complicated. So when you get a rhinovirus infection, ICAM1 is actually upregulated. And dendritic cells, as far as I can understand, are never actively infected, but the binding of the rhinovirus itself to the dendritic cell alters its cytokine release profile. So it comes up with some wacky things like IL-35, which violated my no interleukins above 21 rule. And when they go to the lymph nodes, they become effectively anti-inflammatory rather than pro-inflammatory.
But they don't affect, interesting. And secondly, did you say that there was a single amino acid change that underlies the receptor switch?
There's one characteristic one, which is a lysine residue somewhere. There's other ones that are associated with it. So I don't know for a fact if you made that single change, whether that would be sufficient, but all the minor groups have that lysine. In fact, I know it's not sufficient. All the minor groups have that lysine. Some of the major groups do too. So it's a handful of amino acid changes, not a single one.
And that can switch it from interacting with ICAM1 to the LDRD receptor? I mean, aren't those structures very different?
Totally different. I have not tried to study the
that's very wild.
of it. Yeah, it's totally incredible. People have done experiments in the lab, but like so it's been very challenging to build a mouse rhinovirus model, for example, because they didn't recognize the ICAM1. So you can do experiments in the lab, like throw the viruses into cell culture without ICAM1 and ask them, can you evolve to find a new receptor? And people have succeeded. They get them, I think they got them to use heparin sulfate as a receptor. You know, and of course, there's some cross connectivity between these viruses and other receptors. And I would bet that some tiny percentage of rhinoviruses use other receptors even inside our own bodies. So there's space for the evolution to occur, but it does seem like it's fairly specific.
And just one last thing, at least if you look phylogenetically or evolutionarily, how rapid are these switches occurring in nature?
Probably hundreds of years. So my interpretation of the history of rhinoviruses, which of course is difficult to reconstruct because of their high mutation rates, is they're probably all only a few hundred years old, and that they were spawned by the enteroviruses. Some fraction of things that we call common colds are in fact enteroviruses that come up from your gut to your nose. So rhinoviruses have lost the ability to live at low pH. Their capsid is different. So I would say these things happen on the order of decades.
So this is Gary on. Great talk, Fred. So I'm going to ask a question which may violate some principles of looking at evolution, because it asks for, comparing successfulness of various types of strategies, et cetera. But it would seem to me that the, kind of the fitness basin associated with the major, groups, that's a pretty successful fitness basin, because, it doesn't necessarily invoke a lot of immunologic response to try and kill it, and the things apparently continue to replicate and spread and maintain fairly sufficient populations. So is that interpretation too anthropomorphizing, or is that a legitimate perspective?
It is a legitimate perspective. Yeah, we looked at this because it looks like minor group rhinoviruses are less well adapted. But if you look at the classic studies, which have taken place, since the 70s till roughly the present, the fraction of minor group rhinoviruses is like 15% in infections. I do not understand why that is.
It's John. Fred, the kind of things that Alan's model showed, if the people who were trying to predict cases for COVID had known what Alan's model says before they started doing the predictions with human behavior, might the models have been more useful?
So if you're asking, I'll let Alan weigh in on this too. If you're asking whether the epidemiology models that people used in very high profile ways to predict the epidemic would have been better if we'd understood mechanisms within the host, my answer is no. Actually, I think, we are using pretty crude average parameters of what population level R naughts are. I think Alan's use of the models is important in two ways, right? Obviously to understand treatments better, but also potentially, for what I'm interested in, to understand longer term dynamics with respect to the immune system. But I'll let Alan also weigh in.
I'd agree totally. I think right now, in fact, historically, the link within host dynamics to epidemiological modeling is still pretty weak. There's just so much complexity within host models. But we think the transmission, at least for these types of viruses, has to be reflective of the amount of virus that you have in your upper respiratory tract in some way. And also things like your behavior then play a role. I mean, if one studies the physics of droplet formation, and I'd like to advertise is a great lecture given by a scientist from a National Academy member, in fact, from NIH named Adrian Box, B-A-X-X, and Adrian with 2 A's, he's Dutch, and it was for the American Physical Society. And it's on YouTube right now. And he did experiments. They set up in a lab at NIH and could let people talk in basically with laser reflectometry so they can count every particle. And they showed how size of droplets and transmission depends on if the droplets are coming from skins. So the film layer on your lips that is generating droplets as opposed to your vocal cords. And great pictures of what happens. You add masks and how you can see the large droplets basically become invisible instead of sort of dominating what you're seeing in the air. It's very, very impressive. I recommend it to everyone. But I think that's the one link I have is between what's going on in the upper respiratory tract and it gives something. It's very difficult to, so are thinking about it because it depends not only on what you're doing right now, it's how much virus you're doing over the time interval where you're interacting with people. So you could shed a lot of virus, but if somebody comes in the room a half hour later, if the virus isn't there anymore, it doesn't make any difference. Or two people go by each other, but you're knocking. So there's probably not very much, but if a person goes by you and you shout at them, you look at these videos, you'll see that the amount of virus can go up 100 or 1,000 fold, or the droplets can. So, and how you behavior and put that into these contact tracing models is a whole other level and probably a whole other decade of study.
But I guess R naught is not an intrinsic characteristic of a virus.
That's right.
It's only present in the context.
Right, exactly.
Same is
That's
true
also
for-
true within hosts too.
Yeah, same is
Fred?
true for-
Fred, if your course is available for distribution, I'm sure there are a lot of students worldwide who would love to see it. So if there are any of your lectures or materials that we can share on our website, that would be fantastic.
Yeah, they're all up in an unhidden way on my own website, but I have not YouTubed them. I don't know if there's any legalities to that, but I will check around. They're all up there, but the one when I forgot to hit the record button.
If you could send us a link, we could at least post a link to your site.
I probably don't want to suffer through them. I also put together some lecture notes, but ran out of steam in the middle of the semester, but I'm going to try to finish them.
Okay. Any last questions before we go to back to quick business summary? Jacob, very quickly, if you can, please.
Yeah, Fred, I saw a study that shows that mutations can be used to track things of transmission. It's an Israeli group. Can you comment on this with not necessarily COVID, but other viruses?
Yeah, no, people obviously use it. You know, the viruses are ideal in that they mutate really fast. So there's lots of mutations. My concern, our group has been looking at this actually. There's this idea out there that you might be able to estimate the number of asymptomatic cases by looking at the mutations in known and observed cases, right? And saying if we got from here to here, it must have taken 3 steps, right? I'm very skeptical of that idea because I think even small amounts of sort of asymmetry in contact rates and perhaps more importantly, even very weak selection makes those estimates non-robust. But I do think you can definitely reconstruct phylogenetic trees of transmission. That's quite effective, but I don't know how far you can really go with those.
Okay, thank you. Any last question for Fred? I hope we have opportunity to talk to Alan and Fred extensively through the working group and elsewhere. So I hope this isn't the end of Fred, of our ability to talk. I just wanted to say briefly that we're not having a meeting next week. I hope people don't mind not having a talk on Thanksgiving Day. Jacob and Gary Ann will be speaking December 3rd. Amber Smith, whom we've already heard about from Alan, will be speaking December 10th, probably talking about influenza. Grace, of course, our founding guru, will be speaking. Also on December 10th, we have a NIAID contingent speaking on the 17th, and Filippo Castiglione. Then we're clobbered by Christmas Eve and New Year's Eve. So we will meet then again on January 7th, starting with Paul Macklin, talking about his spatial modeling projects. We have slots available January 7th on. So please send us your suggestions for future speakers. If you want to speak again, that's fine too. We'd love to have ideas of all kinds. Again, doesn't have to be COVID, any viruses, any infection. And it can be not only the kind of in-host modeling we've talked about, but also things like transport, barrier function, droplets. And please help us spread the word about the working group. So please feel free to forward the invitation that we've sent out to you to any groups that you think are appropriate.